another 30 seconds in case anyone come in last minute. Has everyone here all set with the open review? Did you all receive the assignment? And okay, cool. All right, so regarding homework, so the PSET 1, if you're doing it fast, you might have already tried the autograder in the Gradescope, and I think some of the students might have the issue with like autograder not giving you enough the right score. So I think that's mainly a lot of time because in your notebook you need to load the file, which is your training data, and that file has a initial path that is set to your original Jupyter notebook. So if you change that path at some point when you try it in Colab, before you upload it to Gradescope, make sure you change it back to the original folder because that's what the autograder is using to load data. So I think that's just one mistake that the easiest like error pattern that you might run into error. So like just double check you change the path back to the original path that TA gave you in the notebook. And I think you should be allowed to submit a limited number of times to get like a test score, and so that will be the same as the test we're going to use for the final score. All right, so okay, and then again, like once again, if you have any question or run into like autograder issues, feel free to email us and Stefan and my TA. I hope the first homework is not really difficult. It's not supposed to like also like trick anyone. It's just get familiar with the data and get familiar with PyTorch. So yeah, like talk to us if you feel like there's something particularly challenging in the PSET or you have any difficulties. Okay, any questions so far for the previous classes?
Are these notes available on
Yes,
Canvas?
Yeah, they're on module 2, I believe. Cool. So yeah, so we've talked a lot about the DNA sequence a lot in the past. From this class on, for the next about 6 to 7 classes, we're gonna now move on to study protein, which is the next step in the central dogma after DNA transcription. And in terms of protein, today we're gonna kind of learn more about what is protein and what is protein sequence, and what are the kind of the common way of dealing with protein sequences and the intuitions, based on how we're like understanding these data, we can gain some intuition about what kind of model we can apply on this sequence data, right? So we talk about many times about this diagram here, where like protein is the universal like function unit for the molecule that actually function in our body and in the life systems. And the origin of the protein is this DNA, which has this coding region, and it has this exon region where those exon regions got transcribed into messenger RNAs, and then after RNA splicing, the exon will become will then be used as a template to translate into polypeptides, and the polypeptides then, after some folding, have a 3D structure. They become protein, and they have different functions. So during the past many decades, I think that the whole community of like structural biology has this end goal of like using computer program. Like earlier it's like pure computer program, and now it's like using AI to help us understand what happens in the last step of after the protein is translated into polypeptide and before it's folded into protein. So actually, like I guess there's probably 15 years of work on this area, and there are a lot of earlier attempts that kind of gradually build up until like today, or like actually the recent 5 years, where things drastically improved after AlphaFold and diffusion come out. So going back to the basics, what is a protein? So protein can be think of this beads of or continuous or like sequential beads of amino acid unit, where you can see that each amino acid is being because of this translation system happening in our body. We know there is kind of a fixed set of codon, we call it code, or like you can consider it as code, which are of length of 3 nucleotide long. And for each of these 21 amino acid, there is 1 or 2 or 3 such length 3 code that have like 1-to-1 correspondence with the amino acid. I'm pretty sure it's uploaded. Can you try again? Sorry about that. All right, so yeah, so just some basic terminologies to describe proteins, right? When we talk about DNA sequence, we use like amino acid. Sorry, we use nucleotide or NT or base pair to describe each position, or like basically the smallest unit of the sequence. And in the protein, we use residue or amino acid to call the basic unit. So residue means kind of the mostly talk about like this particular location or this particular one amino acid in the protein. And then the amino acid is kind of the unit, the type of fundamental unit that comprises polypeptides and proteins. And as you can see, there are like 4 letters in our DNA, right, TCGA. So there's limited combination of codons. And you can see that some of these codons are pretty similar to each other. They could be like one nucleotide different. So that explains like when you have a small change. So when you have a small change in your genome, you could have a like larger change if that change resulted in translating into a completely different amino acid, even though like in the sequence space they're close. And this can also reflect one type of bias in like how often things are replaced by some other amino acid due to this kind of easiness or hardness of being mutated into something else and getting different decoding outcome. So there are 21 types of amino acid or small unit, or we call it building blocks of proteins. Each of them has a kind of abbreviate as like a single capital letter from A to G. So I think the name is definitely, we don't need to memorize the name, but just to know that there are like this among these 21 types of amino acid, they have some they share some similarities in terms of composition or structures, where each of them is formed by a carbon. There are certain types of atoms that are necessary or always appear in the amino acid, including this carbon, some oxygen and nitrogen group, and hydrogen group. So you can think of each amino acid can be decomposed into, for the most important part, this alpha carbon, or we call it C alpha, which is the kind of the carbon atom in the central of the amino acid, and on the 2 sides of this carbon, so one carbon has consider it has like 2 arms that can form bonds with different like functional groups and other atoms, right? So one of the arm is reaching out to a carbon oxide group, which is on the one side of it, and then the other one is reach out and connected with this amino group. So these 2 groups are fixed. These 3 components are shared across all amino acid, and one of this arm reach out to what we call the side chain functional unit. So this side chain is the only part where the amino acid differ, and the side chain can comprise up to like very small or even no atom or no side chain, or to like a larger side chain that can comprise this like benzyl and bigger units. So and then these carbon oxide group and this amino group. Question?
So my question is, out of the carboxyl group, amino group and the side chain, which one of the three gives it the chemical properties such as polarity, non-polarity, electronegativity?
That's a great question, and we're actually talking about it right now. As you point out, there are different types of chemical properties of the amino acid, and it's the side chain that it's the most determining part of these properties. For example, there is I think we can briefly divide things into maybe four or three types of amino acid. And the most important differentiation is hydrophobic and hydrophilic. So like phobic and philic means like you're not you're not in reaction well with something. So hydrophobic means you're not interacting well with water, or you're kind of afraid of water, so you want to escape away from water. And this kind of amino acid usually have this carbon-rich side chains, which don't interact with this kind of hydrogen-rich water, and it will try to like escape from water. On the other hand, there are also hydrophobic amino acids, or sometimes called polar amino acids, which have like more kind of hydrogen and polarity in the side chains, which tend to form this weak hydrogen bond with water. So you can think of it will be pulled by water, or it tend to like interact with water. They're also like we call charged amino acid, which have kind of this ion that has sometimes have positive and negative charge, and they will also interact with the opposite charge amino acid, or other like small molecules or like metals. So there's also like you might hear people say like aromatic amino acid or something. This also can be like one property depending on whether you have like a benzyl ring in the side chain or not. So we can see that peptide and also DNA is a polymer, and a polymer means like you have kind of repeated unit chained together with same type of reaction. And here the reaction happens between these amino group and carboxyl group. So this side of one amino acid will be attached to the other side of another amino acid, and then the water come out and they combine together. So you get so we'll be able to increase the chain by number of amino acid that's involved in your coding DNA. And and that you can see that this chain also has a direction. Like DNA, we have 5' and 3'. In the peptide or protein, we also have this C terminal, which is carboxyl group side, and this N terminal, which is the amine group side. And we call this this carbon, nitrogen, oxygen kind of chain the backbone of the protein. So it is the single like unique backbone that connect where the alpha carbon is connected together in a unique ordering. And this once this ordering is determined, you have only like unique sequence. So you cannot like shuffle these things around because it's a kind of unique bond that link these amino acid group together. And then you can consider this backbone being kind of doing kind of the overall shaping of your amino acid, whereas this side chain is kind of doing this interaction with the other amino acid groups or other like small molecules or your target ligand or target protein. So these side chains are kind of also in charge of interaction, and the backbone is kind of in charge of giving the over scaffold or shape or skeleton of the protein. So we're not talking about a lot about the structure, but I think in general, like we need to be aware of structure even when we work with sequence. Why? Because structure is fundamentally what determines all these properties and not sequence. So if we consider like this chain of amino acid as a string of beads, so some of these beads might be like hydrophobic or hydrophilic, so they can be water-like or oil-like. So once you drop it down into a solvent, maybe a water, what happens?
The oil-like components stay near the top, while the water-like components, amino acids, protrude into the water itself.
Exactly. So you will see that these kind of biophysics or chemistry will kind of shape, will move the particle around because of different like hydrogen bonding forces, and so that's kind of also one of the driving forces of like why protein has this 3D structure that will largely depend on like the conformation, the composition of the amino acid type and the surrounding solvent. Although like we kind of usually assume solvent is kind of water, or because like I guess 90% of time it's operating in a water environment, but considering water into this modeling system will further complicate the problem. So right now we can kind of set aside water, only consider protein, but we basically know there are some forces that either pulling them away or like pull them together, or just try to compel them or try to attract them. If there's something else that's interacting with the protein, it will also kind of change the shape of this protein a little bit. So you can consider this protein functioning process being this complex energy optimization process where you're starting from like a 3 peptide, and then you've been pulled by these forces and try to like go.
How much variance, or how important is like variance in the structure due to these forces in like practical applications? Because it seems like a lot of models have just considered maybe the crystallized versions of the structure.
Exactly.
But is that super? But like, is that like how much of a gap is there between, let's say, predicting real biological activity and predicting, let's say, the kind of crystallized structure?
Yeah, that's a great question. I think I may talk about it a little bit in this kind of energy landscape, but I think the short answer is there are certain systems or types of proteins that are quite solid. It doesn't move a lot, and it's just like a rock. So and for this kind of interface, we're pretty good at modeling them and designing them. But there are a lot of flexible proteins, like you said, like antibodies, right? They have like this loop region which are relatively elastic, and I think it's still very challenging to accurately model them. And not mentioning the other like things that naturally have like multiple conformations that are also like very hard. Yeah, that's a great question. And then, before we move to the energy landscape, I just want to mention that there are also other forces that I didn't mention besides hydrophobicity, although hydrophobicity is kind of one obvious driving forces of a lot of these floating process. But there could be other, like I said, like if you have like charged residues, you can have this anionic bond. And there's like if you have sulfide in your side chain, you can also have disulfide bound. Which is actually pretty strong force, provide really strong stabilization to the protein. And there could be other like certain like side chain like rotamers where you could basically just rotate the entire side chain to different directions, and that will also result in different energy. So basically, we can consider kind of this folding process or as kind of a traversing of a super complicated energy landscape, where I think most of us believe, based on this kind of Anfinsen's dogma, that there is kind of a global minimum of this landscape, at least like one like super stable global minimum, and it would usually reside into this super deep funnel of energy wells, which means that once you like get inside that well, it's kind of quite hard to escape, and that's why you kind of stabilize and remain that structure there. So that's a lot of the foldable proteins. The landscape will basically look like this. And then we also have additional assumptions like the kinetic, basically the path that you traverse through that space is kind of smooth. So it's not like anything that's discontinued and you need to make like really discontinued jump. Although the kind of the skewness of this landscape can be super, super high. Like you can see this like drastically steep walls that you need to climb up or climb down to get to the optimum. Yeah, so and this landscape is quite, I'd say, yield shape. It has like really like higher order, like changes in the space, and very sensitive to like even small perturbations in the space. But I think that makes this problem challenging and then interesting, and there are a lot of thinking around like how we can find the best way of like smoothing out this space and try to traverse to the most stabilized part of this energy landscape.
So, like the energy landscape would also likely be perhaps determined by, like perhaps the presence of catalysts as well, right?
Mm-hmm.
Like, so the catalyst would would cleave some of the peaks in this landscape.
Yeah, so here I'm only talking about a single like monomer protein, but if you talk about like reactions or involve multiple like a complex system, then yeah, there could be this transition state before you reach like the final like stable state, and then the enzyme will lower the energy of those transition state, so you can get to the lower energy easier. But I think that the stable state will remain the same, the energy of the final like state. Questions?
How often do these assumptions hold?
Well, that's a great question. Yeah, I think I believe the accessibility part it should be somewhat reasonable, but I think the uniqueness might be maybe arguable in my point of view, because we do see things in multiple conformations. But I think if we're talking about truly like global optimum, then I think for this complex landscape without too much symmetry, it's really hard to come up with a second minimum that has exact same energy. So I think a lot of these assumptions are mostly considered like correct. And yeah, that's.
So when you say like sort of this is the stable form of the protein, do you mean that is the stable form in its most commonly observed state, or is there like a uniform state that you're taking the stable form of proteins across?
Oh yeah. So yeah, this landscape, as I said here, like sequence decides the structure and also decides landscape. So this entire landscape is specific to one sequence. Yeah. So for that single sequence, there should be a global minimum state where, for that given sequence, the protein most likely stay in that state. Yeah. But I definitely believe like for some of the type of sequence, the wells are shallower, and it could be, like constantly changing between different conformations. And that's an active studied area, actually, like for how we sample conformers better than, like right now we kind of only model like the single mode of a fold right now. Cool. So yeah, I think the one important thing we want to say here is that everything here is already determined once the sequence is decided. Once we know the sequence, so we can think of the sequence to structure being kind of a sequence to a distribution, right? So condition on a sequence, we have this distribution, which also, of course, as I said, like might be impacted by environment, like solvent molecules, but those can be considered as a conditional variable as well. But like for now, let's say conditional sequence, the distribution of structure is kind of fixed. But the problem is how we traverse this space, how we model this space effectively. So a couple of other concepts that we want to talk about is that there are actually multiple levels of structure that when we talk about structure, and of course the most simple form of structure we call it primary structure is just the sequence information, and it's usually like 1D information, just this sequence of amino acid types. So you will see a lot of like sequence of this thing. Doesn't make any sense to you language-wise, but this is like the most commonly seen form of protein data. And there's a secondary structure where we kind of see that there are certain types of groups formation of sequence, such that if you have this combination of side chain conformation, it will form this very stable helical structures, or if you have two strands parallel to each other, and they're forming kind of also like a kind of internally forming some like hydrogen bonds, that will also stabilize the structure into kind of a flat beta strand. So alpha helix and beta strands are kind of common small, like often observed stable substructures that people usually can use that to annotate like this part of protein is a helix, this part of a protein is beta strand. But of course there are a lot of other diverse type of structures where we do call them like loops or coils that are more flexible and harder to kind of determine, assign this kind of periodical shape. So then this comprises secondary structure. You can also consider it as maybe some type of 1D sequence or 1D annotation, where for this part of the sequence, I just say like this belongs to an alpha helix, this part belongs to a strand. And then so in the tertiary structure, we finally consider the 3D composition and conformation of these different amino acids, where these helices and beta strands and also the loops will then combine together, and they have like the 3D relationship to each other. And then we have this more complex system to model. And finally, the quartary structure, or if we consider kind of a protein, a functional protein being a bigger, big one, like assembly of multiple subunit, and each of them can be relatively independently filled. And so they just kind of combine together to get final functional protein. We may also consider this as like a quaternary structure, and a lot of this kind of dimers or trimers or even non-monomers will have a lot of cyclic structure or asymmetric structures. So this is very interesting and fascinating area as well. And there's also like kind of the notion of domain, where basically the part of your protein structure can be considered relatively self-contained. So yeah, basically if we can consider like this part and this part are kind of folded relatively independently, then they just like finally put them together. So each of these also considered like a domain. So for example, in antibody or T cell receptors, we can have this like antigen recognition part or like antigen binding domain. So these domains are also like sometimes talked about when we kind of want to segment out a particular functional region of a protein. Any questions so far? Great. So although like the structural data is, the structure is what kind of drives all of these complex biophysics process, it is quite hard to directly model structure and learn structure. One reason is particularly like there's like difficulty in experimentally verify the precise structure of proteins in the kind of really high throughput fashion. So realistically estimating there is like only about 200,000 like experimentally solved structure in the protein database, and it's definitely increasing a lot, but it's still like not in the same pace as what we observed in the DNA sequencing area, right? And for large language model, like it's much easier to collect data. You can collect petabyte of data, natural language to train your model, but in protein side, it's not the case. And the CASP is kind of it's called like it's like a challenge, like a Kaggle competition for proteins. It has been hosted since 1994, and people try to come up with like computational, like computer program to try to predict the structure from the sequence from 1994. And I mean, we've been not very successful until like recent decades, where things like with machine learning try to kind of use to accelerate this process. And of course, you can have some like computationally designed protein, or sometimes called de novo protein. And some of them might be like experimentally verified as well, and they got put into PDB as well. So how are we solving this like low data problem? So there kind of mainly got two school of thoughts. Like one is try to directly tackle the biophysics and like using the either knowledge about the complex system or physics. Or like there's part of the physics and chemistry that we know, so we know there are different type of forces. We can come up with empirical like empirical equations for a lot of these forces, and we can directly compute energy. So that's work on these simulation-based method. Let's try to they're applied on the large molecule. They're applied to small molecule as well, and in small molecule you can get even finer grain information. But for proteins, it's simply like really big. You need to model like thousands of atoms together, so it is quite difficult and very challenging. And even the MD simulation here is usually mostly consider a lot of like knowledge-based force field, and they're not really like the accurate kind of quantum level simulation as what we do for like really small molecule systems. And another thing is it's really expensive to do. It's very slow, need a lot of compute, so it really doesn't scale. So the other realm of study focusing on the sequence part of the protein, where actually there are billions of years of evolution that we can borrow some information from, and that comes kind of a study of this homologous structures of closely related proteins, and especially like multiple sequence alignment that we're going to talk about next. so like just to get a high level like understanding of what the data we're dealing with, different from the genomics data we talked about last time, where the proteins are much easier like in terms of the length. They're not like billions of amino acids. So usually a few hundreds to 1,000 is kind of the maximum we're dealing with in this task. And it's also character level like genomics, but we have 21 character instead of 4 character. And besides that, the data, like it's not listed here, but data is kind of small in terms of structure. But the sequence is as much as you can get from genomics, because if you can sequence the genome, you know the sequence for a protein as well, right? So the kind of sequencing, if you look at the sequence data, is about you can get the same scale out of thanks to the DNA sequencing technique. So as a fact, we actually have a lot more protein sequence data. And this data is not just about like the human proteins. We have this protein from all kinds of experiment, and they're like variants, and also like from different species, from species that are like very far away from human to species that are very close. But in general, like interesting finding is that all of these species, we kind of, the theory is we have the same origin. So we believe like a lot of these proteins are inherent from our ancestors, and they are they've been through this kind of millions of years of evolution. And during this evolution, like a lot of the critical part of the functionality of the protein has either changed or evolved to be better, or preserved or conserved, such that the essential function of the life will continue. So this actually provides a lot of useful information about the structure and the function of protein. So so people come up with these thoughts is like, if we just look at all the sequence that we know in the database of protein sequence, is there any rules we can find? Like maybe there are some we call it sometimes active site on the protein, right? Like if you have an enzyme, then you have this particular like small sets of amino acid, and its side chain has this very specific conformation, and it has to rely on this particular part of protein to act to catalyze the key reactions for the reaction to continue. So this part changed rarely changed. If it changes, you can lose this entire function. So there are particularly like conserved residues, and there are also co-evolved residues, right? If two things are contacting each other, if one of it changed, right, it enabled to still have this like contact, whatever kind of bond is forming. You want to change the partner of the proteins to regain that connection again. Right? So this is what we call co-evolving residues, and that has been like really interesting finding that motivate a lot of these later sequence modeling like methods for protein. So the high level thing is here. If we have like a protein that we care about, we usually we can search against a database of all proteins, and there are like very efficient algorithms to search, and then we can pull a lot of sequence that kind of looks similar to the protein that we want to ask about, and we can look at the patterns on this set of sequence. We call it multiple sequence alignment. And yeah, I forgot to mention the contact map. Contact map is also kind of a close grain information of the 3D structures. So we consider just like a binary mask, binary matrix of like between all pairs of residues. You can also assign distance to this matrix, which is more finer grained information about like pairwise distance. So it's basically like a graph. It's an adjacency matrix of the graph defined by these like protein amino acids. And the contact map prediction has also been a very interesting task in CASP, and it's kind of the step before predicting 3D structure. Are we able to predict contact map? And that has been like challenging, but we can see that like this contact map tells us about also like co-evolution, or actually co-evolution might have correspondence with this contact map, because like things that are contacting are like more likely to co-evolve with each other, and therefore there's kind of a the covariance between these two positions might be different from the two positions that are completely independent to each other. Questions?
What percentage of proteins have sufficient coevolutionary data to
That's another great question. I think for a lot of the training data for in the PDB that we used, they're at least able to come up with, a reasonably deep MSA. But for a lot of kind of like the disordered region, which are really hard to find any homologies, for those like type of disordered region, it just doesn't work. And like another reason why you might not be able to find homology is you kind of doing some de novo design, which completely go outside of the natural distribution of protein sequence. But otherwise, that we should expect kind of like commonly appear like natural protein should have some homology.
You had shown on the protein data bank 200,000 proteins, which seems to suggest that was small. Were there
That are experimentally verified.
And so I would think with all the data and whatnot, we couldn't. What's the issue that we can't find more?
The issue that we cannot predict more or we cannot test more.
Oh, I see. So those are been tested.
Yes, tested. So we can predict millions of proteins already with AlphaFold, but like whether or not it's correct is debatable. For even for AlphaFold 3.
Wet labs.
Yeah. You need either kind of X-rays or using like cryo-EM electron microscopy to get the information. And also those cryo-EM are super noisy and have a very expensive to get. Cryo-EM. Yeah, and I'm sure we'll talk about it maybe in the structure modeling module. And another challenge of like not being able to experimentally verify is to be able to verify it. You need to have it in like a soluble or stable state. That means a lot of times you kind of crystallize the protein, and not all protein are like easy to crystallize. And there's like a lot of these structural wet lab experiments work on previously. They have to think of different way of maybe adding salts or adding different things to crystallize it, and so we can get a stable structure.
What is like AlphaFold, for example, like made it easier because you know what structure you're looking for, as opposed to earlier you would have had to try like quantum different approaches. So has like the timeline to verify protein structure like shrunk since then?
You mean did AlphaFold accelerate the verification? Maybe, yeah. I think that's an interesting question.
Because now you know what you're looking for. You have a good idea of what it's supposed to look like, and therefore you have
I think
to narrow down on what sorts you want to add.
Yeah, I think in terms of design, like we definitely test more things that are more meaningful right now than previously. Like previously we don't know what would work, but right now like with AlphaFold we have some level of like use that as some level of filter for things that at least like fold or stable, so that drastically increases success rate for like designers like like David Baker's group at Protein IPD. They've observed like a huge increase in success rate, even though success rate is still like maybe below 10%, but previously it's kind of like 0.1% or 0%. So it's quite a big leap. Cool. Yeah. So now let's I think we need to talk about multiple sequence alignment. So what in a high level, what multiple sequence alignments is, we try to like gather the sequence by this kind of a similarity to the query sequence, and we also try to align them by positions, such that we can infer what happens in the evolution. So just before some details of MSA, I want to mention like this MSA information is heavily used in the AlphaFold, at least AlphaFold 2, and AlphaFold 3 is still using it. And it is actually the biggest part that consumes a lot of compute and parameters, even though the predictions is on the single sequence. So this MSA is really helping, like providing this information about contact, which actually cannot be inferred just from sequence alone. And they found that like if you remove this MSA and only put a single sequence to fold like natural proteins, which are like long and like really complex, it's actually not very successful for AlphaFold. So usually, like if you don't give it MSA, you have like this result, which is like much lower than if you give it some MSA information. And people try to like understand what's so important in MSA, right? So they try to like basically, there are two ways of using MSA. Either you look at the column-wise distributions, and there comes this PSSM we're going to talk about. So if they try just feeding the column-wise distribution, assuming independence between positions, they still cannot get like a good amount of information. And if you also like assume kind of row-wise information, if they do shuffles of columns, then it still don't give you enough information. So it's really this co-evolutionary of positions that kind of it cannot be modeled by this independent model that is giving these signals.
So I guess just looking towards the future, I guess this means this really restricts our capabilities in terms of using AI models for predicting protein structure to proteins where we have like this evolutionary data. So that means no like out of distribution, new to nature proteins or new amino acid sequences and things like that.
Yeah, I think. I mean, that's a great question. I have like two things. One is we actually found, interestingly, if you're not looking at the natural proteins, which are big and complex, if you look at the design proteins, which are really short and usually very well shaped, a lot of helices and beta sheet, the prediction is pretty accurate, even without MSA. So that's kind of like a sweet spot where when you try to go outside of distribution, but you land in some distribution where AlphaFold is very confident about. So yeah, I think somehow like the designers, when we do this kind of diffusion and generative model, we luckily landed up into a region that is somewhat like confident by AlphaFold. So even without MSA, it is able to like fold it correctly.
When you say designer proteins, like what types of designer proteins would be the common examples that we might like observe or study about?
Over what?
Like what types of proteins are designer proteins that we might be looking at or study about?
Yeah, we're definitely going to talk about any kind of approaching the end of the semester. There's so many types of protein that are being designed right now, like from just like small binders, right, binding to some targets or enzymes, right?
Small molecule inhibitors.
Yeah, small molecule inhibitors. If you know the small molecule, or if you know like even large molecule like receptors, if you want to design a binder that binds to it, it's really simple right now. I mean, it also depends on what type of binding. Like I said, like solid interaction is much easier. And there's also like if you use it for antibodies, which are more challenging, and you can use it for like ion channels, even design like membrane proteins or like nanopores or cyclic proteins, all kinds of fancy but like nice looking proteins. So yeah, I think. But I think it's a great question. Again, like I think there would be a time that that we have to go outside of this nice niche, good distribution to actually reach the real functional protein that are more complex, even without MSA information. And that's it's a very interesting topic that like even me myself is interested in doing research on. So I believe there should be like more generalizable way that MSA information can be carried to a single sequence. But we're kind of actively investigating that. So now let's talk about MSA finally. Basically you can consider MSA being this kind of a process of aligning different sequences to get the consensus of them and understand what happens during evolution. This is just saying that not just protein have this evolution, but also like other substructure like RNAs or like regulatory motif that we talk about in the module 1, right? But we can see that the evolutionary pattern are quite different, the signature, right? Because they are the mechanism behind this evolution are just different. For protein, you have to translate it into amino acid and then doing this interaction with the amino acid side chains, whereas for regulatory motif, the DNA directly interact with the protein, right? So the kind of the interaction here is different, and therefore the evolutionary signatures are also different with these different types of data. So So that's why the one model may not fit all. Yeah, so why we need alignment? Because during this course of evolution, a lot of things can happen, right? So the simplest thing, which will save a lot of our efforts for a line, is mutation. So if like one thing changes to another thing, then it's easy. Like you have like almost 90% of sequence the same, and you know, we should be varying only by one amino acid here. Nucleotide here. But there are more complex things happening because you can have deletions, where like this base pair will like disappear, and then the A will become like directly before G, right? And you can also have insertions where something like random nucleotide is added into your genome. So this makes the alignment more trickier, right? So basically the multiple sequence alignment or alignment process is to infer what kind of added operation has been done in this process from like one sequence to the other. I mean, I think the higher level the higher level goal here is to kind of first we want to define recover what kind of operation happens under this defined sets of operations: insertion, deletion, mutation. And we also want to define some criteria of how good the alignment is. Also, we need to define some scoring function. And then we want the algorithm to be run like efficiently on the large sets of proteins. If we do things brute forcefully, it would be terrible. So, I mean, this can be like if you have taken CS466 questions.
This is more of a biology question. Can you go back to the slide with the deletions? Doesn't like deleting base nucleotides from the sequence shift the entire reading frame and switch the whole protein? How are they still homologs that they're producing?
Yeah, it's possible that if you get a frame shift, you can have like entire domain dysfunction. That's also possible, yeah.
Okay. So is it common to see homologs that have so in homologs what kind of mutations do you most see? Do you see like base pair switches or deletions? Is it like what proportion of mutations, deletions, or insertions do you see among homologs?
I think so definitely mutations will be more common than gaps. Gaps is either insertion deletions, but you would see a lot of gaps as well. So yeah, I'm not a expert in homology, but if you're interested, definitely there are two other courses offered by our faculty, like CS466 is offering this semester, and CS591 offering next semester, taught by Tandy. They're the expert of sequence alignment. And yeah, I think it's a great question, and there are a lot of things we can learn from observing what happens, under this timeline of evolution, and try to figure out who's the ancestry, who's the child. So yeah, I think I'm just going to briefly talk about the sequence alignment, since there are other two classes you can go to, but just to give you a sense how complex this problem is, right? So the easiest thing to do is kind of substring matching, where maybe in your algorithm class you've learned about it, right? So if you wanna find the longest common substring, no gaps, just the one substring of these two, what do you think it would be? All right, I'm not gonna trick you. I think I'm pretty sure you can do it. It's the basically GTCA, right? So then you've offset it by two. Then the longest substring can have is GTCA, but it's really a small portion of this protein, and this might not be the optimal one if we consider that there could be gaps, right? So when we actually there's better formulations so instead of finding the longest common substring, we find a subsequence where you can allow some gaps in between the sequence. So therefore, if we kind of allow the break between A and G, and then you can match this A with this A here, and then skip this C here, right? So basically, the longest subsequence we're getting is much longer. It's 6 instead of 4. Furthermore, we know that when we introduce these gaps, and we can also sometimes introduce some mismatch, right? For example, in this case, C and G, they're just not aligned together, right? So they're not common sequence, but they're still paired. So there could be a mismatch happening, which may be suggesting a mutation from C to G. But we basically want to define some cost when we introduce this kind of error or gaps or mismatch. So then there's the final formulation of sequence alignments. Basically, try to come up with cost functions that score this like mismatching or deletion or gaps with some level of prior knowledge about like maybe how likely that mutation is happen, or like whether or not we want there to be such a gap or mutation or not. And there's like a lot of background on like how this score is setting up. But basically, we can have like the understanding of like for all pairs of amino acid into a sequence, you can give it as confidently the score. So now when we consider gaps, then the sequence alignment becomes kind of the problem of gap placement problem, where you basically want to, the longest non-boring alignment, meaning like you cannot have gaps at the same position because that's meaningless, right? So if you have the constraint that you have at most one gap at each position, then the longest non-boring alignment is n plus m, right? So if you're placing gap in different position, you basically try to like set up with different way of like matching the sequence. So we can think of this game as being like decide to place or not place a gap at each of these n times m entries. So in total, there could be 2 to the n plus m number of solutions, which is kind of an exponential space we don't want to deal with. So then there comes the algorithm part, which is kind of people can find like super smart dynamic programming algorithm, where we would break down this problem and solve it iteratively with recursion, such that we can drastically bring down the compute time from exponential to polynomial. But the brief idea here is we're kind of considering this kind of matrix of matching scores. So each pair of the positions here means the best cost you need to pay to match the sequence up to this part of the first sequence and this part of the second sequence. So the intuition here is you could break the longer matching problem into a matching of smaller subsequence, where like if you assume that you have basically solved the previous all possible matching patterns before adding C and G, you could pick the best one of them, and then do the operation that will add in C and G alignment, right? And then you can also add in the score that you need to pay when you do this additional adding or will not add, whichever we decide to skip it or we don't skip it. So we will update the score for the next alignment with the next position. So yeah, this is the really high level intuition of dynamic programming here. So we enable to compute the best score we need to pay to match at a different part of the protein, the sequence. We kind of just want to kind of fill in this matrix from left corner to the right bottom part. And once we find this best score, the best alignment will be just the path that we travel from, the beginning all the way along the matching step that gives us the best score, all the way to the final, like the right bottom corner. And to be able to do this traversing, it also costs another like O(n) time algorithm to do it. Yeah, so this is a kind of illustration of like how this is doing. Like you basically try to use dynamic programming to fill the scores, which will automatically solve like also like a path where you can like backtrack to get the alignment. Yeah, I think there are ways to accelerate the algorithm, so you don't need to consider like things that are too absurd, like the routes that are too absurd. We just don't we don't need to want to further explore and compute anymore. Yeah, so that can further accelerate some of these algorithms. So yeah, so if you take Hamid's class, you will see kind of this kind of 2D like finding the path along this 2D matrix. Which is finding the best alignment, and multiple sequence alignment. So far we're talking about like pair sequence alignment, and there's like it's always a challenging problem, but just looking at pairs does not give us coevolution, right? So effectively be able to see that covariance we need more than 2. So there's multiple sequence alignment where it's essentially the same problem, but you're now dealing with a larger chunk of set of protein sequences, and of course, kind of then everything kind of needs to generalize to k sequences of 2 sequence, And again, we need to kind of define some scoring functions for K sequences, and the kind of alignment now extend from like a 2D matrix to, like KD space, KD hyperspace. Okay, yeah. Again, like if you're interested in these algorithms, there are like fascinating research going on this side for the other two faculties in BCB area, and feel free to take these two classes to understand more. So assume we have this sequence alignment, right? Now we want to actually look at patterns that will impact its function, right? So in the earlier days, we have this kind of sequence signature that we can define, where I think basically here the end goal is still like to extract some patterns, such that for example, if we want to search against like a set of new sequence, we know like for the new sequence whether or not they belong to this set of sequence, or that they have similar pattern with this family or not. The other thing is we can use these signatures to also like maybe make predictions of the function itself. So in the earlier days, we come up with like really easy way of summarizing these MSAs, right? So the easiest way is to kind of just find, common amino acid. Like for this position, A and C is the most abundant one, so I just like say A and C is the kind of signature of this position. And take it one step further, we can compute the probability of different amino acid at different position after we align it. Right, this is very similar to the motif we found in DNA data as well. It's exactly the same thing, like the precision weight matrix, except that we compute it on the amino acid base. And we can also use these small motifs to kind of, as a fingerprint, to scan like the longer proteins, right? And we have multiple motifs, and then we can decide like the combination of multiple motifs as being like the signature or something. And finally, we can build a probabilistic model, right? Assuming that each of these position has a probability defined by some PSSM, and you can have like a hidden state that is decide like, right, do I want to switch to another state such that I have a different distribution at this position? So assuming kind of some like independence between or conditional independence between these positions, we get this simple like probabilistic model defined by PSSM. If you kind of want to get some level of confidence or measurement of whether the sequence belongs to something or not. However, we want to like, however, this model can be like overly simplified because we assume this independence among positions, and the joint distribution can be much more complex than like K independent distributions. And particularly, we talk about this covariance, right? So a pair of residue and how they co-evolve is very interesting and important. So we want to also capture this correlation information between different positions. So there are also studies that, families of models that try to like study whether there is independency or correlations between two positions. Right? You can look at basically the information gain if you consider them as joint or independent. And this can be one metric. And you can also, using this metric, you can compute neutral informations or other like statistical coupling similar to like how much information basically how confident we are that these two positions are co-evolving and not independent to each other. But still, this is not like a probabilistic model, right? If you give me a sequence, I cannot give you a likelihood just using this statistics. So then people also try to come up with graphical models which try to give you a likelihood, basically try to model the probability over a set of sequence. Graphical models are kind of a very flexible family of probability models, which you can define some like independence or conditional independence or correlations. So Potts model is one of such graphical model that try to specifically capture this pairwise correlation. So it's kind of a graphical model that defines some energy between all pairs of amino acid, and they also consider like a single amino acid as part of its energy. So basically, it's an energy-based model where the energy is computed by looking at every pairs of amino acid and every single amino acid, and you can train this model using a lot of this Bayesian modeling methods. Which we might talk a little bit also if we'll do like variational autoencoders later. But one interesting methods to like learn this Potts model is using kind of a restricted Boltzmann machine, which is kind of a early days deep learning, where you could consider like factoring these like visible variables in the sequence into some hidden variables, and by doing this factorization, you kind of have a more conditional independence where you can run MCMC more efficiently than running the pos model directly. But yeah, I think this is still like a well thought after model that captures the as much as we can from kind of a prior design angle. What if we just want to do it totally like data dependent? We want to do it totally data driven. We don't want to come up with these graphical models because our graphical model can be wrong, right? So that kind of inspired the actual machine learning or deep learning over the multiple sequence alignment, where maybe starting from AlphaFold, which use these deep neural networks on top of MSAs, to also like some later transformer architectures that directly define on top of MSAs, and we're using very simple formulation where we kind of you can give it a task that I give you a set of MSAs and I mask certain positions of the MSA. Can I predict or recover this masked position? So instead of doing masked sequence modeling on a single sequence, we can do it on a set of sequence, which is kind of like the flavor of this MSA transformer. And they also have like particularly designed attention mechanism that is try to cover this co-evolutionary by basically designing specific attention or axial attention that looking at the column-wise and row-wise patterns. And EvoFormer or AlphaFold is another very successful example of running deep learning directly on MSAs. So similar to transformer MSA transformers, it also has several layers, deep layers on top of these MSAs, and it also have this axial attention, which look at the column-wise and row-wise patterns. But after that, I try to combine it with this pair representations, which is so instead of considering just the number of sequence by L is the length of sequence, we also explicitly model like interaction between different residues. So we have these pair representations of L by L, matrices, which basically you can consider like outer product of each position, or each residue in your MSA with the another residue, which gives you kind of this interaction patterns between two positions. So in this EvoFormer, we're gonna talk about it more during folding and the second sequence modeling class. But in a high level, when we look at this pair representation, which can be considered as a kind of some level of adjacency matrix, or even like a coarse, a vague prediction of contact map, then there is some geometry that we want to respect if it's a contact map, right? One such geometry is the triangular inequality, right? So if you have these pairs of edges from 3 nodes, and forms a triangle, then you know that the length of these two should always be like longer than this edge here, right? So in able to keep this kind of trigonometries, they come up with this triangular self-attention or triangular update attention that actually looking at every triplet of the element in this matrix instead of every pairs, like the normal transformer we're using. And they come up with like a very specific way of doing information passing or message passing, or you can say attention with masks, that try to kind of aggregate information such that we are sort of aware of these triangular constraints. And that makes the pair former, even though it's kind of a model over sequence, very powerful, and also being able to capture some like a 3D or co-evolutionary information. So yeah, I think this is a really good example of like how we should think out of the box of natural language, because in natural language you kind of rarely do this pairwise thing, right? You kind of consider everything as sequential, and you believe like your syntax is like enough, this serialization is enough to capture all of your information. But in the protein, we actually found that the pairwise information, or looking at it in the 2D space, is more important because of this coevolution. And there's more like geometry we need to be aware of, even though we're modeling with sequence. And finally, MSA is kind of what we've relied on in the past, where we try to, by hand curate these evolutionary information together, and then just like run model on top of them. Like, what if we further just let the model itself to figure that out? So I think evolutionary scale model, or ESM, is kind of one of such leading methods in the direction we call protein language models, or even like maybe some people will call MSA transformer as protein language model as well. But I want to particularly separate the two, because this protein language model is really a language model on like a single sequence of protein, and it just follows whatever natural language processing does. And the intuition here is all of the sequences you use to come up with MSA, they are in the database already, right? So if you kind of if you learn the joint distribution of the entire database, you also learn the joint distribution over the MSA. And with the, protein language model, the belief, especially with the mass language model, is that it's trying to capture the joint distribution of sequence, especially with the transformer, which are kind of less, sequential and more kind of like equivariance to different positions. The belief here is with like mass language modeling, we're fitting the distribution over the entire protein space, such that we're also fitting on this, joint distribution of MSAs, and such. Therefore, we have some level of coevolution information as well. Question?
So, I read the assignment. There's a paper to the side. They are trying to develop a DNA vocabulary. So I haven't read some paper about trying to build the protein language vocabulary. So is it impossible to build a protein language vocabulary?
You mean like tokens for proteins, right?
Yeah.
Yeah, I think that's a good question. But on the other hand, I think amino acid is a really good vocabulary of protein already. As you know, that it's an entire like subunit of your protein, which you can't or cannot break down anymore or make it more complex anymore, right? Because certainly I definitely have reviewed paper that try to do tokenization of our proteins. But I myself, I don't believe that it's necessary. It's particularly when we deal with like hundreds of amino acid. It's not like genome, which is super long. You have to tokenize it. And secondly, we know that the DNA, you have these kind of small local motifs, which are in charge of interacting with like transcription factors, right? So you can't break those up. These motifs are really fixed. But I think protein is much more flexible. You can easily piece together very different. Yeah, but all the de novo design we do, like we try to come up with new combination of amino acid. Although I agree that there could be some smaller patterns, like Hamers, that, for example, if you like have this consider this structure of helices or beta strand, and of course you need like certain types of side chain conformation. Therefore, you have like fixed, like maybe consecutive like 3 mers or 6 mers that can give you a helix. But that's not very interesting because like helix only have, I think most of the time helix is just like stabilizing, the protein. What's interesting is in a lot of these loops that are, flexible and precise, and also like in this really precise local conformations that can come from multiple, distantly located amino acid, and you cannot consider them as token because they're like very long distance away in the protein because of this 3D folding. Yeah, questions?
So the optimal slide, I saw the edges are directed. A lot of statistical models that you show, like false model and correlation, they're not really directed, right?
Yeah.
So were there some advantage by introducing these directed edges?
Yeah, that's a great question. But I think for the POTS model, there isn't really a direction, right? We only care about correlation. But here, we care about the kind of geometry. So I think the reason they have direction here is they kind of want to break the symmetry, such that like if you go from ij to, so the edges coming out of j and coming out of I are dealt differently. So you have some of asymmetry in the attention.
Did it help a lot?
Yeah, definitely. I think they did a lot of ablation study from AlphaFold to AlphaFold 3, and triangle attention is what they cannot remove from AlphaFold 3. They remove a lot of other things like exo attention, like the one that Tanya said, like look at the row and column wise, but they have to keep the triangle attention or triangle update for AlphaFold 3 to work. So yeah, I believe this. I think this is also this direction directed. In this direction is also to fulfill the triangle inequality, as I mentioned, right? You have to let it know that these are the two edge of the triangle I'm looking, and this is the edge that should be always smaller than these two edge. And you have three sides of this triangle, so you have basically three way of arranging the arrows, right? So then you have both directions. Again, this is not a graphical model. This is more like to keep the geometrical constraint. So it's a little bit different from Pod's model. It's not an energy-based model, and it's not a graphical model. Cool. So yeah, pretty language models. Like I said, so ESM basically is one such earlier effort where ESM2 only use available protein sequence in the database, which is I think about 680 million sequences, and they just do simply mask language modeling, where you randomly mask part of the protein and let it recover it. And then at the end of this pre-training, they actually take out this high-level embeddings or attentions, which is also pairwise, interestingly. And then they put this attention into a folding trunk, which is very similar to AlphaFold. And then they see how much information they can gain from this pre-training. Would they be able to directly predict some information in the structure? And so surprisingly, they actually recover that, like even with the zero shot, like with no fine-tuning, directly take the attention out of these pre-trained models, it actually already reflects some 3D structure, especially in terms of like contact map. So if they it freezes. But I think they've done like zero-shot prediction of contact map and structures using this attention map, and they were able to actually recover quite a lot of it. And the other thing is they actually take these high-level embeddings as a pre-trained features for proteins, and they use that, fine-tune that to directly predict functionality of proteins, such as binding, or for antibody binding or immunogenicity, or some stability. And they actually find that this embedding is pretty predictive of certain types of protein functions, which are closely related to the structure of the protein. But interestingly, I think someone recently did another study that they fine-tuned ESM embeddings for a wide range of functional predictors, and they found that for functions that are maybe not directly related to structure or not uniquely determined by structure, the transfer learning performance is not as good as like functions that are like more rely on structure. So it's kind of pretty obvious that the ESM model is learning more about this structure contact in the way that we talk about, like being able to model sequence database as a way of model MSA. So yeah, I think these are pretty much what I want to talk today, and I think we're a little bit early, so I'll give everyone 2 minutes back, and if you have any questions, you could also ask right now.
So is the MSA was like almost pre-processing in the olden days, and now it's become like a role of the transformer. The transformer does the MSA transformer does the MSA now, or?
Not really. I think people still believe in MSA. I mean, there are people still actively use MSA and are suspicious about ESM, because ESM could be memorizing some information about MSA, so it cannot generalize to new sequence, right? So like even if you memorize MSA well, doesn't mean you generalize well. Like it doesn't mean you're actively learning the interaction in some degree.
Was the MSA the old version, whatever, was that a learning task?
Not really. MSA is solved by the algorithm, dynamic programming.
So why did they have an issue if now they say
Yeah, so we can consider dynamic programming or the